everyone. So, in today's session, we are discussing a question that has appeared in the Life Sciences GATE question paper of 2020. This is question number 18 from the biochemistry section. So, this question states that a PCR in 100 microliter reaction volume containing two primers at a concentration of 0.2 micromolar each is set up to amplify a 250 base pair DNA fragment. Consider the average molecular weight of one base pair as 660 Dalton if the primers are fully consumed by the end of the reaction. The amount of the final PCR product formed in microgram. Okay, so in this question has come from a basic technique which is called PCR. Okay, so PCR, or we can also expand it, we call it polymerase chain reaction. Okay, so polymerase chain reaction is to amplify a certain fragment of DNA from the whole sample. Okay, so if you have a particular sample and from there you want to amplify one particular gene segment, okay, so to increase that copy number of that particular gene segment, so that you can proceed with that particular region. Okay, so if you have this huge sample, okay, and you want only this much fragment, okay, only this much fragment, so after the PCR you can have that particular fragment in abundance. You will have many copies of only that particular fragment after the PCR is done. Okay, so that is the work of PCR. Okay, so let's talk about the basics of the PCR. Okay, so the components of the PCR. So the components of the PCR are: number one is the template DNA. Okay, it should be a template DNA which will be denatured in the beginning, okay, where the primer will bind. Okay, so this template DNA could be anything. It could be a genomic DNA if you just want to amplify one particular gene using the genomic DNA. Okay, from the genomic DNA, if you want to amplify a gene, there your template is genomic DNA, or you can have the template as a particular plasmid. From a plasmid, you want to amplify one particular segment of the plasmid, then your template DNA will be a plasmid. Also, if your particular gene of interest, okay, gene of interest harbors intron in it, and you want to amplify the fragment without including intron, what you can do, you can isolate the RNA from the organism, convert it to cDNA, and now this cDNA, which is devoid of introns, you can use this cDNA as a template to amplify any particular gene. Okay, so these could be your template DNA. Okay, so from the template DNA, to amplify your desired fragment or desired segment of the DNA, you need to design the specific primers for the annealing. Okay, so primers are required so that it can bind after the denaturation of the strands, and because our polymerase can extend the 3' end of the primers, so it needs a 3' end that is free, and with the 3' end the polymerase will keep adding the complementary base pairs. Okay, so to provide that 3' end we design primers. So primers are generally 50 to 25 base pairs long. We should have two primers, one forward primer, another reverse primer. Okay, so we'll be having forward and reverse both the primers, and this is the usual length. The TM, the melting temperature of the primer, should be in range of 50 to 70 degrees Celsius, and for the annealing we calculate by deducting 5 degree from the TM. Also, there should not be the difference. The difference in TM should be less than 5 degree between both the primers. And for the strong binding of the primer to the template, we always try to keep 40 to 60% GC content of the primer. And these are the requirements for designing the primers. Other than primer, we also need DNTPs, that are the building blocks for the next or the newly synthesized DNA strand. So it will include all the deoxynucleotide triphosphate. We need a thermally stable polymerase, and we need a buffer with trivalent cation that is important for the enzyme activity. So these are the things which is required for any PCR reaction. And we use, so in generalized way, if I'll say, we can use 1 to 100 nanogram of our template DNA in any range according to the what sample we are using for the as a template. Okay, primers we generally provide 0.1 to 1 micromolar range. Okay, the DNTP we provide the final concentration of DNTP we try to keep generally 0.2 millimolar. Will the tag polymerase, that we generally 1 unit, 1 to 5 units are good enough for PCR reaction. The buffer that generally we have, if we have 10x concentration or we have a 5x concentration of this buffer, we make it to our final 1x concentration. Okay, so these are the requirements, general requirements which we use in the PCR. Okay, so so in our PCR reaction, which is a 3 steps. Okay, so there are 1, 2, 3, 4, 5, 6 steps. The first one is this initial denaturation. Initial denaturation. Okay. And then we'll have the denaturation step, small denaturation step, because we need to repeat the cycle. We have our annealing step. We have our extension step. We have our final extension step. And we have hold after the reaction ends to the stored. And this extension, denaturation, annealing, and extension. This step we repeat 30 to 35 times. Okay. So these are the steps involved in any particular PCR. Okay. So we generally starts a huge piece of DNA, to amplify one particular short gene of interest. So that has certain length, suppose 1 kb length. Okay. And uh, this is how, using the specific set of primer according to the how many primers we are giving, okay, our desired fragment will be amplified. Okay. So these are the components and these are the PCR steps: denaturation, annealing, and extension. So our question which states that a PCR of 100 microliter, okay, so a 100 microliter reaction is being put, okay, so containing two primers at concentration of 0.2 micromolar. Okay, so they are using 0.2 micromolar primers. Okay, so whatever uh, whatever concentration of primer you will provide here, that many fragments will be binding. Okay, so if you are providing like one, two primers, if you are providing, you will have at the end two molecules. Okay, if you are providing 10 primers, so they will give you 10 molecules. Okay, so accordingly, if 0.2 micromolar primers are provided, our end product will be 0.2 micromolar DNA. Okay, 0.2 micromolar DNA will be formed. So it is set up to amplify a 250 base pair DNA fragment. So considering the average molecular weight of one of the pair is 660 Dalton. Okay, so it has been given, like suppose one base pairing. Okay, so this is one base pair. So this one base pairing molecular weight is given, which is 660 Dalton. That we should know. Generally in the competitive exams, sometimes it has been provided to you the weight of the one base pair, or it needs to be known what is the weight, molecular weight of one base pair. So if one base pair, the weight is 660 Dalton. If it is only one single nucleotide, and then that single nucleotide will be 330 Dalton. Okay. So it is written, if the primers are fully consumed, so whatever you are giving, they are being fully consumed in the reaction, so the amount of the final product formed, it is being asked to you. So as we are saying that if we are providing 0.2 micromolar of primers and it is 100% consumed, okay, so you will have 0.2 micromolar DNA formed. Okay, so the answer it has been asked to us in microgram. So let's convert molar to micrograms. Okay, so what is a molar? Okay, so the molar is number of moles per liter. Number of moles per liter. Okay, so number of moles are nothing but weight by molecular weight per liter. Okay, so we know this molarity. So the molarity which is given to us is 0.2 micromolar, or we can also write it 0.2 to 10 to the power minus 6 molar. Okay, now weight which is in gram. So weight we need to find out, that we'll be finding out. So molecular weight will be, so for one base pairing it is 660. So there are total 250 base pairs. So it will be multiplied by 250. Okay, in 100. So the volume is 100 microliter. Okay, so if we intend to convert microliter to liter, it will be 100 into 10 to the power minus 6 liters. Okay, after solving this whole thing, we'll be getting a number that is, okay, okay, or solve it like this. So this much gram. Okay, and the answer which is being asked to us in microgram. So we can also write as 3.3 microgram. 10 to the power minus 6 can be converted or can be written as microgram. So the answer would be 3.3 microgram. Okay, so the basic simple equation you need to know first, because the number of amount of primer whatever it is required, okay, that many DNA fragment will be formed at the end, because it has been asked in micrograms. So that molar you have to convert into the grams. So what you should know, what is a molar? A molar is number of moles per liter, and number of moles are weight upon molecular weight, and molecular weight you can calculate if you have known the amplicon size and the base pair molecular weight. And using that entire calculation, you can end up getting the DNA amount in microgram or gram. Okay, so this is the solution of this question. Thank you for listening. See you in the next video.